All right, yes, indeed. Thank you so much, Thomas and Laura, for a very interesting review of the state of the art or state of our ignorance in the field, because there are many question marks, many advances, but also many question marks. And the bias we took in the group over the past 5 years is actually to go back a little more as far as we can to the mechanisms that could actually enable the phenomena of connectivity, functional connectivity, resting state connectivity in the brain. So let me explain a little bit basically the approach. Basically, what we are looking at different scales, like we've seen very nicely earlier this afternoon, are these complex signals, either at the surface of the scalp or with LFPs, depth electrodes, or source imaging in MEG and EEG. And because these signals are complex, we're interested in a possible effect of experimental manipulations due to behavior or pharmacological modulations of these things. So just to make things a bit simpler, we imagine that we can actually modulate those things with an external event, like it's represented on this cartoon. And the classical approach so far that we've seen today in some of the talks, just as a matter of review of the state of the art, is to represent average outcome of, for instance, measures of event-related responses, behaviorally related or pharmacologically related to these aspects. But we can also introduce further dimensions. So time, yes, frequency, of course, and power and phase representations. So everyone has seen, I guess, in papers or in their lab, these kind of representations. The problem when we do that is that we know that the signals are basically built from oscillations. It doesn't need to be rhythmic. It could be arrhythmic, like we've seen also this afternoon, but they are manifestations of oscillatory phenomena. So there is a mechanism here that where different rhythms or bursts of oscillatory activity coexist, but we pretend we are agnostic to this phenomenon, and usually when we look at for instance, time frequency representations of the average signal, we usually look at band-limited effects, pretending that the rest of the spectrum is not actually sensitive to the manipulation in question. So this is not very satisfying. And the bias, again, that we took in the lab, like I said before, is to consider the relative robustness. I put that between quotes, of course. There's been beautiful talks today in other rooms about the difficulties to actually reproduce and actually robustify, so to speak, the outcome of fMRI resting state analysis. But the case study that we looked at in the lab is actually how to bridge the gap between these apparently robust observations of bold fluctuations in the resting state, yielding those resting state networks that we've all heard about and read about, with the apparent complexity and diversity of the expressions of the resting brain in electrophysiology, from manifestations of rapid and low amplitude bursts of beta oscillations to relatively slow and high amplitude oscillatory activity in the delta range or even lower than that. And because the rationale was that if we are able to bridge that gap during rest, well, then we would be very well equipped to tackle the difficult problem to look at the transition between rest and task performance and or behavior. So that was the roadmap, the game plan that we came about. And to do this, because again, the inspiration and motivation was bold, observations of bold fluctuations and correlated fluctuations in the resting brain, we looked at the literature, which is relatively thin, but very, very interesting, that looked at, coexisting or concurrent recordings of bold activity with electrophysiology. So this is only one example from Dave Leopold's lab. I'm trying to look for the mouse, and it's trying to escape me, but that's okay. So on the y-axis, you have the typical spectrum of electrophysiology. Forget a little bit about the x-axis. It's just the map that you see here is a cross-correlogram between the envelope of the typical oscillatory components in electrophysiology and the bold time series that are measured concurrently here in the monkey brain. And whenever you see red, it means that there is a strong, substantial correlation between electrophysiology and BOLD. When you see blue, there is actually anti-correlation, meaning that whenever electrophysiology is strong, BOLD is relatively low. And what was striking to us is that actually the whole spectrum is actually engaged, so to speak, in this correlation across modalities. So you see strong positive correlation from high gamma to low gamma. So this is basically a nice reproduction of Logotiti's works, seminal work at the turn of the 2000s. But also interestingly, you see at the bottom of the cross-correlogram very strong correlation with the other brain rhythms, delta, theta, and alpha, and which was not observed originally by Logotitis for, I guess, technical reasons regarding the access to low-frequency electrophysiological components. What is also very interesting is that the beta rhythm is actually also related to BOLD, but in an anti-correlated manner, meaning again that whenever beta is strong, BOLD tends to be low and vice versa. So it's good news in the sense that indeed there is a good, at least in the basic dynamical aspects of the slow fluctuations of these oscillations, there is a relation with BOLD, but it doesn't tell us much about the mechanisms. So we have to go back to the blackboard, so to speak, and try to basically look at possible interdependencies between those brain rhythms. Because with maybe the exception, and it's limited, of the beta range, there is very few evidence that any of these typical frequency bands in electrophysiology yield the resting state brain networks that we observe in BOLD. So instead of looking at all of these frequency bands separately, and it's not only true for, again, resting state, it's just a case example, very telling, I think, but it's just an example. We hope to be able to actually extrapolate and generalize to actually the active brain during task performance. But instead of looking at these typical frequency bands separately, we would like to see whether there are possibilities of interdependence, like the Satupalva just mentioned before. Why? Because if these different brain rhythms coexist, there might be two possibilities. Either each rhythm is basically a fingerprint of different subnetworks of the resting state. It doesn't seem to be the case so far as the literature can tell us. And also during the resting state, whenever the brain is active, and it's active, as you know, all the time, well, these things are present, you know, with different expressions of power and relative amplitudes, but they are always present. And there must be mechanisms why they coexist, and there must be a fingerprint, a signature, a structure in the scaffold of those brain dynamics that we can probably look into to extract and simplify signatures of resting state brain connectivity. So one possible key is to look indeed at cross-frequency coupling, like it was said before. And again, it's going to be a little bit of a repeat, but it's just a cartoon that illustrates one very well investigated mechanism of cross-frequency coupling, like it was said, which is phase amplitude coupling, or PAC. Why is it interesting? It's because it's actually a possibility, there is the possibility to bridge the different, again, expressions of true oscillatory activity and arrhythmic bursts of activity at different frequencies together to make, basically, to have a mechanistic picture of the expressions of the dynamics of the brain. So PAC, in a nutshell, is basically the, like again it was said before, the relation between the phase of a relatively slow oscillation with, like here, that we're going to call the frequency for phase, with bursts of faster oscillatory activity that we're going to name a frequency for amplitude. So there is a repeat or a consistency of those bursts with the phase of the slow oscillation. Not only is it interesting to put everything in the same picture, even though here we're looking at slow versus faster, that it's not a continuum in the spectrum. I'll come back to that later. But there are also physiological aspects that are interesting because it is believed from basic electrophysiology that slow oscillatory activity, which is relatively high amplitude, can be in most cases a signature or a shadow of a group or net excitability of the neural cells or the neural assemblies that we are basically measuring from with LFPs, but also with MEG or EEG source imaging. So it's an interesting signal extraction technique that brings us actually relatively closer to the functional aspects of physiology, which was kind of missing in our perspective in the recent past. So I'll come back to that later. So to put things more into perspective and add to the picture, so to speak, so let's consider actually it's a tentative approach to a holistic view of the electrophysiological spectrum and the different possible roles of the different frequency bands. So we've seen fast and slow brought together with phase amplitude coupling, but what about the intermediate frequencies, in particular the beta range oscillatory or bursts that seem to be so prominent in some aspects of the brain activity, but seem to be anti-correlated with the rest of the spectrum. So this is just a proposition that I think has the only benefit to bring together all the possible frequency bands that are typically observed, and also to allow us to propose operational hypotheses, testable mechanisms with experiments and of course analytics. And I will show examples of that in the second part of the talk. So let's consider a basic neural assembly or cell assembly built from typical excitatory and fast and slow inhibitory cells. And again, it's just a simplistic view, but we have to start somewhere. Like we've seen in one of the previous talks, these things, under certain circumstances that are physiologically plausible, can express spontaneous phase amplitude coupling between slow oscillatory activity, again, which is a shadow of their net excitability or levels of excitability, and faster bursts of oscillations, essentially in the low and faster gamma. Well, whenever there is an input to the system, it could be a stimulus towards primary sensory areas, but also at higher order levels of the networks in the brain, one network being the input of the next one. Well, there is a certain processing that is happening at this initial level. I will come back to that later. But what's going to happen for sure is a propagation of this input signal towards higher order brain regions, just to simplify. This propagation can be either direct, but it is believed that a great deal of those bottom-up propagations are relayed by cortical hubs, but it could be also subcortical network hubs like the thalamus and other structures. And as you know, a great deal of perception and perceptual mechanisms are actually involving prediction. And what we think happened, or what could happen, is that the top-down mechanisms actually, or the top-down manifestations of these predictions could be conveyed by beta oscillations in a timely fashion. So if we indeed move to the time domain, we need to imagine that indeed there are fluctuations of net excitability just by the physiology of these neural cell assemblies. During these resting state phases, there are spontaneous expressions of phase amplitude coupling between low frequencies up to theta, maybe alpha, and gamma oscillatory activity coming in bursts at certain phases of these slow oscillations. And whenever there is a stimulus or an input to the network, there is a certain form of phase resetting that happens so that the predictability of, for instance, just to simplify, the timing of occurrence of the next expected stimulus is actually predicted so that the system is in the best possible position to actually register the next incoming stimulus. That's one possibility in the time domain, but we can extrapolate also in the spatial domain and other higher order, even cognitive maps of perception and action. So that's just, again, a roadmap that we propose to test in a very systematic fashion. And the only benefit here, I think, even though it might appear a little bit simplistic, is that it brings together, again, the whole spectrum of electrophysiology into one place, where each frequency band, typically observed empirically in electrophysiology, has a certain role that is related to function, physiology, and maybe higher order, of course, aspects of cognition and behavior. So yes, like I said, the second half of the talk will be to put that into an experimental perspective at different levels. So I'd like to go back to a relatively old experiment we did with Karim Gerbi and colleagues where we looked at visual motor integration. Basically, the task was relatively simple and consisted in basically manipulating a trackball, as you see here, to play a very simple video game. And just to relate ongoing, and this was, as you may imagine, it was a block design where for several seconds the person was actually manipulating the trackball. And the difficulty was actually to relate this ongoing brain activity during task performance with the behavior. And you have to remember that until not so long ago, it was a little bit taboo to look at spontaneous brain activity in a non-event related fashion. And it's only relatively recently that we let ourselves relax a little bit about this and take a leap of faith and actually pretend that ongoing brain activity is not noise, but it's actually very rich in terms of spatiotemporal dynamics related to behavior. This is only one example out of now a very large literature in electrophysiology. So what we did is look at long segments where the person was playing this video game, capturing behavior by looking at hand velocity. And when we looked at basically spontaneous M1-like contralateral activity, there was no evident correlation except in a specific frequency band. And this is exemplified here by filtering both signals and by overlaying the two time series, and you see a very strong synchronization, if not coherence, between the two. Of course, this can be done in a non-exploratory fashion. You can look at coherence between behavior and brain activity, and it's not a form of connectivity, it's a form of behavior to brain correlation, so to speak. And if you look at the spectrum of the hand movements, there was a peak on average at about 2 Hertz. If you look at the spectrum in the brain activity, rest is in green. You see the strong resting, idling, basically rhythm around 10 Hertz. There was a small but significant difference at actually a slower rhythm around 4 Hertz. And when we computed the coherence between the behavioral signal and the brain signal, there was indeed a peak, very strong peak at 4 Hertz. So as a manifestation of coherence between brain signals and behavior, which is, I think, important. And then you can proceed in the same manner by actually going step by step and now look at cortical coherence. And you take this M1-like region as a seed. And again, the mouse is disappearing. That's OK. And basically, you compute systematically the coherence with the rest of the cortical signals, and you find SMA, more parietal regions, and also cerebellar, ipsilateral cerebellar regions. So that was the basics of basically relating this oscillatory activity with behavior. And now I'd like to share with you a very recent study that we did with Benjamin Morillon in the lab, who is now a PI in Marseille. And it's basically looking at the possibility at this, again, at this predictive aspects in perception, here in auditory perception. And Benjamin was actually interested in looking in the possible engagement of the motor cortex in auditory perception. And our hypothesis is that the accuracy of the temporal prediction of the occurrence of an auditory stimulus would be conveyed by this beta burst I was talking about in a top-down fashion. So the task was basically a pitch discrimination task over a melody that was played at a 1.5 Hertz rate. Yeah, this is an example. That's the standard tone. And then the reason why it sounds faster is because you have a target melody that is actually played at the same tempo as the first standard notes that are played at the beginning. And then intersped with the target melody is a distractor, basically. So you have to keep track of the exact timing of the occurrence of the notes when it is expected from the reference tempo, hence these strong expectation signals that we thought were conveyed by the beta burst, all right? And at the end of the day, the participants have to report on whether the target tones after the standard tones were actually played at a lower or higher pitch with respect to standards. That's just details of the experiment. And basically there were two conditions. In one condition, they were allowed to tap with their left hand at the tempo of reference, and we wanted to verify if the actual engagement of the motor cortex would yield better performance. And indeed, it's been shown by Benjamin in other behavior paper before, but we confirmed this finding by actually reporting on enhanced performance when the person was tapping along the tempo versus just listening. And so to analyze, to report again on possible effects related to oscillatory components in brain activity related to behavior and performance, we looked at stimulus to brain connectivity or interrelationships by basically fitting a 1.5 hertz sinusoid to the standard target melody and comparing the brain responses to the 3 hertz intermixed melody of target plus distractors and trying to look at the differences. And we looked at the stimulus response or stimulus brain phase locking. And indeed, not surprisingly, when you look at the 3 hertz components in the brain that are phase locked to this 3 hertz sound track, so to speak, you see very strong involvement of the left and right auditory plus cortices, as shown here. Now, if you look at the 1.5 hertz tracking, correlation between the behavior and the brain, what you see during the tapping or tracking condition, and again, not very surprisingly, you see a strong involvement of the right sensory motor hemisphere and sensory motor cortex, sorry, and because again, the participants were tapping with the left hand. So far so good. It was essentially for quality control. Now we tested specifically some elements of this mechanistic model I put forward briefly before by looking at the possible engagement of or participation of beta bursts that would be actually occurring within locked to the phase of the 1.5 hertz target tone. And indeed, so what we did is looking at this possible model that whenever, for the target melody, there would be some processes in the brain and possibly in the sensory motor cortex where these beta bursts would be occurring in phase with the expected timing, of the target tone. And we looked at it in the tapping condition, and indeed in the tapping condition, you see again, like I said before, this engagement of the contralateral sensory motor cortex, but also of the ipsilateral sensory motor cortex, which was less expected. So here, this is a plot. The map you see is a plot of the phase amplitude coupling of beta bursts, possibly occurring in phase with the oscillation at 1.5 hertz. And in the listening condition, where there is no explicit sensory motor activity and behavior, what was interesting to us to observe is indeed the engagement of the sensory motor cortex. And when you look at the conjunction with the tapping condition, this area on the ipsilateral cortex of the hand movement is actually present here. And actually, so we looked at the phase locking with, of the sensory motor cortex with the rest of the brain and looked at performance effects. And indeed, the more the person was actually in phase with the expected stimulus, the better the performance and the more the engagement of this inferior frontal gyrus as well. So you see, by actually putting forward a possible mechanistic hypothesis, you can actually, one possible benefit, even if the hypothesis is wrong, is at least you start from somewhere. You build a block or a game plan not to get lost in the exploration of your data, especially in the different frequency bands that are expressed. Now this is also data from the lab where we also tested the possible contribution of these beta oscillations in the predictive power of the stimulation. And we took one paradigm from Pascal Free's lab for testing visual attention, where basically we showed contracting rings at a certain rate, here 3.5, 3.4 hertz, 3.2 hertz, of optical flow. Okay. And the task was very simple. The participants needed to press a button whenever they thought that the rate of contraction was changing. Importantly, we had different phases in the design of the experiment. So first, we started with a pure resting period of time, where before the experiment started, to capture some of the resting state brain dynamics as baseline. Then we had the stimulation period per se, but between trials, we had this base or baseline, if you will, segment of several seconds that would precede every single trial. And when we looked at the event-related expressions of oscillatory activity, not surprisingly, we saw a great deal of induced gamma by the visual stimulation. That was actually the motivation in the first place why we selected this experiment, because we knew from Pascal Free's lab's results that it would induce this gamma activity. Our interrogation was whether this gamma was actually tonic or whether it was related to underlying oscillatory activity, especially in the lower frequency range. And indeed, when we looked at the activity in the visual cortex, we indeed detected induced gamma, like it's shown at the bottom of this figure. And when we looked at how this gamma was related with a low frequency activity, this is a comodulogram of potential candidates of frequency for phase versus frequency for amplitude, we saw that indeed there was a great deal of activity, of gamma activity related to the phase of the slow oscillation. So the apparent continuum of gamma activity during stimulus presentation is actually not exactly a continuum, but these are bursts of gamma that are phase-locked to the slow oscillation. So the next thing that was interesting to us was actually to confirm this by comparing with resting activity. And what was statistically significant in terms of phase amplitude coupling is indeed this gamma to low frequency at 3.2 hertz. Stimulus-driven slow oscillation, but also the presence of beta activity, as you can see here at the bottom of this figure. Yeah, no luck with this. And what was also intriguing is that the comparison between this baseline segment pre-trial, that were preceding every trial, with the resting state. So no stimulus was present during this baseline period, but only the expectation of the next stimulus to occur. And what we found is indeed phase amplitude coupling of beta activity with the expected timing of the stimulus, timing in the sense of the dynamical aspects of the stimulus. So this could be manifestations of these top-down expectation signals from higher order, maybe attentional circuits towards primary visual areas, because this is all coming from primary or occipital visual areas. Now what we would like to do is actually change the predictability of this stimulus by changing the low frequency of the stimulus in a predicted or not predictable fashion. I have 2 more minutes, I think. And I would like to finish with this recent study that we published earlier this year, where we actually established a causal role of these oscillations in performance. Because again, we are observing, different modulations of oscillations, but it's not for certain that these oscillations are not just epiphenomena of brain activity with functional aspects that we cannot actually probe with MEG, source imaging. So what Philippe Albouy in the lab did is actually a working memory auditory task by playing simple melodies, like 3-note melodies, in 2 segments of time. The first segment was just the presentation encoding of the melody, and with a retention period of several seconds, and then the same melody or some altered basically versions of that melody were played eventually, and the subjects needed to realize or tell us whether the note in the middle was changed, the pitch of that note was changed. And there was a difficult condition where actually the melody was played reverse, to engage even more, manipulation in working memory. So there is a simple task, and a difficult condition, and difficult condition is the melody played backwards. And indeed, the performance of the subjects is better in the simple condition than the difficult one. I'm going to skip this. We looked at the IPS region, which has been documented from functional MRI, but also EEG and MEG as being strongly engaged in working memory processes, especially manipulation of objects in memory. And we saw indeed that there is a stronger involvement of that region during the difficult condition. And when we looked actually at the conjunction between the correctness and the difficulty of the task, there is this strong engagement of these left IPS regions and other regions a bit more prefrontal. And there was simultaneous EEG recorded, and essentially we found the same with EEG, which is always good to see, I guess, just for confirmation. But more interestingly, we saw also during the retention period stronger expression of theta oscillation in that region. And the theta oscillation was actually, the strength of the theta was actually related with the performance of the subject. So the more the theta you had in that region, the better your performance. So the question came out that if we were to entrain that theta by using neuromodulation techniques, would that actually have a positive or negative influence on the task, on the performance? And that would therefore establish a causal effect of neural oscillations on performance. So that's what Philippe did. So we used the TMS over this region in every subject and in a rhythmic or arrhythmic fashion. The rhythmic fashion was designed to entrain the theta oscillation and look at the resulting behavior. And the arrhythmic was actually some form of control condition. What we observed, as seen in the middle here, is that in the difficult task, in the difficult condition, there was improvement of the behavior of the performance for arrhythmic TMS and no improvement for arrhythmic TMS. And we looked at connectivity. I think it's the next slide. Oops. I don't think I have time to show you anyway. But we looked at connectivity and also the strength of this theta. The theta was actually stronger in those who improved the most, and the connectivity within brain regions, IPS and prefrontal networks was also stronger for those who improved the most. And I'd like to finish with just 2 slides on connectivity per se with the resting state, just to close the loop with my initial slide. And what we would like to propose is that a phase amplitude coupling model of resting state dynamics could help reveal the resting state networks for the reasons that I was trying to explain, which is that these phase amplitude dynamics might be related to the intimate modulation in time of basic physiological function of these brain regions. And a couple of years ago with Esther Florin, we proposed the model of synchronized gating, which is a bit of a generalization of the communication through coherence model by Pascal Fries. And what we did is exactly reproduce phase amplitude coupling measures like Bob Knight did in ECOG data. So actually we use this code to estimate phase amplitude coupling. We looked at specific time points just to simplify and downsample the time series in the resting state at every voxel of the brain, and looked at the amplitude of the gamma, connected the dots, and created a surrogate, if you will, or simplified versions of the MEG source time series, which actually spectral dynamics is very similar to the resting state bold, peaking at 0.01, 0.12 hertz, and then applied the typical extraction techniques of resting state networks used in bold fMRI. And what we found are basically typical resting state networks spontaneously expressed in the MEG resting state data, but not from a data-driven, not obtained with a data-driven approach, but with this mechanistic hypothesis of conjunction of phase amplitude coupling dynamics. So I would like to finish, and thank you for your patience. With just take-home messages is that neural oscillations, as I hope I summarized today, but also with the great talks we heard earlier today, are not noise. They are not just epiphenomenon of brain activity. You shouldn't be shy about looking at them even in the resting state. It's actually a very rich source of possible signals and extraction techniques that you can relate to behavior and conditions if you are looking at patients. Even very sophisticated measures like phase amplitude coupling, when you think about it, these are very tiny statistically recurrent observations of little bursts of high gamma activity nested in the phase of slow oscillations. The SNR is actually quite weak, and there are caveats for measuring those things, indeed, like we saw before, but they are accessible non-invasively with MEG and probably EEG as well. So this is good news. And these neural oscillations, you can untrain them to establish causal aspects, causal relationships between their observations and behavior. And I think it's important to now try to have a more complete view of the whole dynamics in the brain rather than looking at specifically delta only, theta only, alpha only. I think it's important to at least take the risk, maybe not in aim 1 of your grant application, but maybe aim 2, to look at in an integrated fashion at all these dynamics. And I think now it's time to actually take that leap of faith and use these mechanistic models. So I want to really thank you and thank my lab members for their work and your attention today. Thank you.